with Parkinson's disease. And so interest was piqued, and a lot of effort and time went into trying to understand why it could be that this gene called GBA could seem to have a role in Parkinson's disease. So this is another bit where it gets a little bit technical. I'll try and keep it as plain as I can. But basically, as I mentioned, genes produce proteins, and one type of a protein is an enzyme. In fact, probably one of the most common types of protein that's produced in our body. So you've probably heard of enzymes. It's the same stuff that you get in your biological washing powder. And that enzyme's role is to break down molecules into so that they can easily be washed away in your washing machine. So it converts big molecules into smaller molecules. And that's exactly what the enzyme GBA does in the body. It takes a big form of fat and creates a smaller form of fat from it. And it's sort of like a dustbin, really. It's within the cell. It's something that just gets rid of used up products from the walls of the cell and just gets rid of them into a safe form. But what became quite clear with people who carried these mutations is that this enzyme wasn't working the way it was. It wasn't breaking down the big molecules into the smaller molecules the way it was supposed to. And so people started thinking, well, actually, if we could find some way of getting that enzyme to work in the way that it would if there wasn't this mutation, could we start to be thinking about the possibility of a treatment that might have some effect on stopping the progression of Parkinson's disease? Because of course we have drugs which are very good at treating the symptoms of Parkinson's disease, certainly in the early stages anyway, but we really don't have very much in the way of treatments that slow down the progression of Parkinson's. In fact, we don't have any. So a lot of effort went into it and a lot of time was spent. And this is where there is a great deal of advantage in having a gene which is responsible for a very rare disease and a very common disease. Because it means that you can use research that has come straight out of the rare disease and just plonk it straight into the context of a common disease. And in fact, Gaucher disease has been known about for about 40 years, and a lot of very, very successful research has gone into it. And one of the things that they did was they basically took what's called a drug library, so about 4,000 different drugs, blindly selected, didn't know what any of them did. And what they did is they added each one of them to some of this enzyme to see if it was able to increase the activity of this enzyme, so increase the amount that it's able to turn the big molecule into the small molecule. And they went through all these compounds and they found one drug, completely out of the blue, absolutely no suggestion that it would do this at all, which did this. And that drug was called Ambroxyl. You probably haven't heard of Ambroxyl. In fact, has anyone heard of Ambroxyl? So it's a cough medicine. If you go to Germany, everyone in the room would put it up because they dish it out in bucket loads. You can buy it over the counter. It's sort of the go-to cough lozenge. But no, in fact, so we had an MSc student at the lab who was German. She thought, oh, Ambroxyl, yeah, I've got some at home. We'd never heard of this stuff before. But it's a cough medicine. The thing that's really great about it, it's been used safely in Europe for about 30 years and has really very little side effects. So the only side effects that it produces are that it gives you a little bit of tummy upset. Some people get a bit of an allergic reaction to it, and that can be a bit worse than with other drugs. and it can cause a bit of reflux and a bit of nausea sometimes. But other than that, the drug was really, really safe. And what's really excellent about that is that when you develop a new drug that comes straight out of the lab, you have to go through quite a long process. You do what's called a phase 1 trial, which is you find somebody who really needs money quite a lot, and you give them a tiny, tiny dose of it, and gradually increase the dose, so that, and then you look at their liver, and you look at their kidneys, and you look at all the other things, and you try and figure out, and then you make sure that there's nothing toxic about the drug. And before that, you have to do lots and lots of animal studies. So you give mice ever higher doses of it, and then monkeys, and oh gosh, it gets in. And then after that, you do what's called a phase 2 trial, which you take a small group of patients, and then after that, only then can you go to what's called a phase 3 trial, which is when you actually test whether the drug itself works, not that it's safe, or that it does what we predict it does in the lab in real humans. Only after all of that can you progress to the point where you do a clinical trial where you're actually testing whether the bloody thing actually does what it says. It actually slows down Parkinson's. And the average cost of development of a drug pipeline from molecule in the laboratory, phase 3 trial, is well over $1 billion. And takes over a decade. But of course, if a drug's been in safe use for 30 years, then you can skip, 70% of those steps, and you can really just progress to the point where you're actually starting to, actually see whether it first of all, whether it does what you think it does, what you predict it does in the laboratory in humans, and then secondly, whether it actually works. And so, to cut a long story short, that is, well, actually no. Let me talk about one other thing first. We developed the sort of context of how it came to pass that this drug Ambroxyl became a potential drug for use in slowing down the progression of Parkinson's disease is due to the story that we sort of just talked about. But what's really, really exciting about Ambroxyl is that there is a lot of evidence to show that even if people who don't carry mutations in this gene, this GBA gene, they have a reduced level of activity of this enzyme. So if you look at the brains of people who have Parkinson's disease and compare them to people who are the same age, who don't have Parkinson's disease, the activity of this enzyme is reduced. The same in the fluid that flows around the brain, and also in a variety of cell models of animals and other things which we know have a predisposition to Parkinson's but don't carry this GBA gene. So there's a lot of hope that not only will this drug be of some use in people who carry abnormalities in this gene, but will also be of use of people who don't, because we think and we hope that the mode of action that leads to Parkinson's is similar. And so if that is the case, then giving this drug may provide some kind of therapeutic benefit. And so that brings me to the Ambroxyl trial, which started in 2017 and finished in April of this year, of last year rather. And it's taken, I'd never run a clinical trial before. I didn't quite realise how much effort it is to actually do it. The amount of paperwork and bureaucracy and governance that you have to go through. But finally, we have the results, and the paper has been submitted. And I can't tell you what those results show, but they are quite encouraging. I should be clear that the trial that we've just done is not one of these phase 3 trials. So it's not a trial where you're looking to see if the drug actually slows down the progression of Parkinson's. what we're doing is we're looking at people who have Parkinson's disease, giving them the drug for a certain amount of time, taking a sample of the fluid from the back of their spine, which flows around the brain, and looking to see whether we can duplicate things that we've seen in the laboratory in human subjects. But there is, interestingly, a larger trial going on in Canada, which is due to finish soon as well, which is actually looking to see if it can test whether the drug has an effect on the progression of some of the features of Parkinson's. And interestingly, they are looking to see whether it can slow down some of the memory problems that occur in Parkinson's disease. The reason they're doing that is that people who carry this gene seem to have an accelerated course of some of the memory problems associated with Parkinson's than people who don't. So it will be very interesting to see what those results come, and obviously we don't know what they are because the trial hasn't finished yet. So that's where I'm going to leave that. But what I am going to just talk about briefly, if time permits, is to talk a little bit about what the implications of this kind of approach are to Parkinson's, and where we think, treatment for Parkinson's is going to go. It's abundantly clear that people who have Parkinson's aren't the same. In fact, they're completely different. There are people who get it very late on and have a tremor and lead tiny amounts of levodopa. There are people who get it very early in life and progress quite rapidly. And then there's the whole spectrum of people in between. There are people whose only symptom is a tremor. There are people who don't have a tremor but have lots of stiffness and slowness. There are people who disproportionately fall over. And I could carry on. And I'm sure you guys are much more aware of that than I am. But we treat Parkinson's like everyone's the same. Everyone is seen at the same time, at the same interval. I'd like to say 6 monthly, but unfortunately that's just not true. Not in Devon and Cornwall anyway. We give the same treatments, more or less, to everyone. And it just seems to lack a bit of nuance. It just seems to lack a little bit of kind of understanding of the disease process. Because what really we should be doing is trying to isolate the people we think are going to progress most rapidly and then target therapies at them. And the other thing is that what's very, very interesting about patients with Parkinson's is that, and I'm sure many people will be able to relate to this in this room, that about a decade before they got Parkinson's, there were very subtle signs of Parkinson's, which you probably didn't pick up, but in retrospect, and certainly if I go through them, you may well think, oh, hang on a moment. So, people with Parkinson's tend to lose their sense of smell about 10 or 15 years in advance. People who develop Parkinson's often have interrupted sleep in the decade or so before. They thrash around at night. They have horrible nightmares. People who develop Parkinson's often are very constipated in the decade or so before the diagnosis of actual Parkinson's is made. And this is, on the one hand, not very good because the symptoms are not nice. No one likes not to be able to smell a glass of wine. No one likes to be sitting on the toilet for hours on the day. But on the other hand, potentially, these are quite useful things from a clinical perspective because they open the prospect of actually being able to give that if you come up with a drug that is able to slow down the progression of Parkinson's, then you might be able to give these drugs in advance of when somebody gets Parkinson's. So when they've lost their sense of smell only. And one thing I can tell you that was quite exciting about our trial, it was not something that we were looking for, is that when we were taking when people were taking the Ambroxyl during the trial, two people who had absolutely no sense of smell, about 3 months into the treatment, regained their sense of smell, which was not something we were expecting at all. And in fact, we really wished we had, because we would have actually formally tested their smell at the trial and at the end of it. But these kind of symptoms are, in themselves, extremely non-specific. Anyone who smoked will likely have not a very good sense of smell. Somewhere in the region of about 20 to 25% of the population are constipated to some degree or another. Sleep problems are very, very common. Actually, the sleep problems you get in Parkinson's are quite specific, but in order to diagnose those properly, you need to sit in a room with all sorts of wires on your head for 24 hours. And so doing that for every single member of the population is not going to be feasible. But what might be feasible is looking to see if there's a cluster of those symptoms. So somebody who's got poor smell, who's constipated, and has really poor sleep. And then the thing that you can add into that, which is even more interesting, is people who have these mutations. So I've talked about GBA, but there are other mutations that are similar, none are as common as the GBA mutation, but there are other mutations which around there which have a role in Parkinson's disease. Some of you may have heard of LRRK2. Has anyone heard of that? Yeah. So LRRK2 is a lot less common. About 1% of Parkinson's patients in the UK carry LRRK2 mutation. But actually, if you think about the number of people in the UK who have Parkinson's, that's actually quite a significant number. And the other thing is, at the moment, if we look at what's called the heritability of Parkinson's, so how much of Parkinson's can be explained by having a family history of Parkinson's, we estimate using some very, very, and it's not me by the way, this is other people much cleverer than I am, but they estimate that about 70% of the heritability of Parkinson's is unaccounted for. And so the hope is that there are other genes, like GBA, like LRRK2, that are lurking around there, that we just haven't found yet. And one of the reasons we haven't is the limitation of the genetic techniques that we have available to us, and I can talk a little bit about that with people if they would like. But actually, it may be possible if you have, for instance, somebody who's lost their sense of smell, who's quite constipated and has very poor sleep, and then has a mutation, say, in the GBA gene, to say, well, this guy is a sitter for Parkinson's, let's give him a drug, even though we're not 100% sure he's going to develop it, because the side effect profile is quite good and it's cheap, and actually, in the long term, it's going to make a huge difference in his quality of life if he doesn't get Parkinson's. But conversely, it's going to save the taxpayer a lot of money as well, because the cost of nursing homes and all of those sorts of things that go with Parkinson's, all the medical care that goes with it, is quite substantial.
It's stopping coughing as well.
Well, do you know what? It's very funny. I said I wouldn't talk too much about the trial, but there was a guy who had bronchitis during the trial, and they asked me, he said, how, you notice much difference with the Ambroxyl? He actually said, no, I haven't really, but I tell you what, my cough got a lot better. and the reason for that was actually because we're using a dose which was about 10 times higher than the dose you normally give for cough medicine. And the reason for that was from studies done in monkeys. That was basically that was the dose that we realised you would need in order to get it into the brain. But yeah, he loved it. And I guess the point I'm going to end on is just to say that the cost of genetics has come down dramatically. Genetic testing has come down dramatically. So in the 1990s, the 100,000 Genome Project, the first genome cost somewhere in the region of $3 billion to genotype. You can now genotype your entire genome, and there are companies that do it for around about $1,000, and that's going to fall even more. And the reduction in costs of genetic testing are actually going to mean that this kind of population level screening, you looking for genes that are predisposing people to Parkinson's disease, and then combining them with other measures, like some clinical measures we talked about, or maybe the kind of DAT scans or PET scans that some of you may have had. These things are actually becoming a possibility. And so the hope would be that if Ambroxyl does turn out to be a viable treatment to slow down the progression of Parkinson's, or indeed if other treatments, and there are other treatments that are targeting the that same GBA pathway which are not Ambroxyl, and drug companies are investing heavily in this, by the way. So it's not just it's not just us. There are a lot of people putting a lot of money into this. That actually this kind of approach of using genetics to guide treatments, personalise your treatments in Parkinson's may actually become a reality. So I guess that's probably where I'm going to finish. I've talked a little bit about the journey of the change in our understanding of Parkinson's from a non-genetic to a partially genetic, and I would say predominantly genetic disease, although that's quite controversial. Talked a little bit about how the slightly unusual story by which this gene GBA was identified as having a role in Parkinson's disease. And I've talked a little bit about how we are using that information from these genetic studies to start to tailor treatments to target those genetic pathways. And then a little bit about how this may impact on the future. And I guess the point I'm just going to finish on is that I'm sure you have all sat in this room and been mightily sort of cynical about people like me coming up and saying that, and we think one day that we might find a cure, sort of in very vague terms. And it's something that we certainly have been guilty of. And I'm not saying that this is going to happen overnight. There is probably going to have to be a few more clinical trials coming up. But I've never seen a situation in the field of Parkinson's where the volume of research going into drugs that slow down the Parkinson's disease, and also the results that the preliminary results that are coming through have been so promising. So I really do hope that there may be some respite in the next few years, and actually that we may actually have at least some degree of treatment that may be able to slow down the progression of Parkinson's disease in the next 5 or 10 years. And that's what I'm going to say. Thanks very much.